Hello and welcome to all my Gulf students. So today I'm going to start with the ultimate recap of the chapter Principles of Inheritance and Variation Beta. This chapter is dealing about what are genes and how these genes are inherited from the parent to their offspring. So basically you all know that genes are the unit of inheritance, right? And this whole concept, the basic concept was explained by Gregor Johann Mendel, who is also considered as father of genetics. So basically in this chapter, we are going to study about the details of genetics, right? So first of all, beta, how do you define the term genetics? So basically, genetics is a branch of science that deals with the principle of inheritance and variation, right? Inheritance and variation, or you can say with the it's a branch of science that deals with the pattern of heredity and variation. Heredity is simply inheriting the characters from the parents to the offspring, and variation is the degree of difference between the parent and the offspring. And it is all because of the sexual reproduction, right? We all are the products of sexual reproduction. We are not asexual products. Hence, we are variable from our parents. We are different from our parent. We are not identical. But still, there is some similarity between you and your parent. It all lies in the hands of the pattern of genes which you receive from them. Correct? So what and how will you define the term genetics over here? Chalo, let's write. So basically, beta, it is the branch of science that deals with the pattern of heredity and variations. Right? Clear? Father of genetics is basically Gregor Johann Mendel. He was the first one to explain the pattern of inheritance of gene. Right? Now, question arises in our mind that what is a gene? How will you define a gene basically? So, beta, gene is actually referred to the basic unit of inheritance, which is chemically made up of DNA, right? These genes are located specifically on the chromosomes. Each gene has their own specific location on the chromosome called as locus. And these chromosomes are then present inside each nucleus of the cell, which you actually receive from the parent, right? So how will you define genes basically, beta? These are the unit of inheritance and chemically they are DNA, which are located on specific positions on chromosome called locus. Specific of chromosome, yes beta, called locus, right? Now about gene, about the pattern of inheritance, the first scientist who tried to explain was not Gregor Johann Mendel, but some other scientists like Knight and Gauss, etc., who due to some or the other reason could not get the success, the amount of success which was received by Mendel himself. So basically, beta, Mendel worked on garden pea for 7 consecutive years, and he chose 7 contrasting characters, each represented by 2 pure line, a homozygous pure line dominant and a homozygous recessive pure line. And with the help of those 7 characters, Mendel basically tried to obtain monohybrid cross and dihybrid cross. And on the basis of the results of monohybrid and dihybrid cross, this fellow Mendel proposed certain laws which explains the pattern of inheritance of gene, right? Clear to everyone? Chalo, very good. So basically, the next point you're going to write is that Mendel worked on garden pea. The scientific name of garden pea is what, beta? Yes, very good. Pisum sativum. Right? For when you write a scientific name, then both underline them separately. Right? For 7 years and chose 7 contrasting characters. And chose 7 pair of contrasting character for his experiment. His experiment is basically hybridization experiment. Now those 7 characters are, so let's write the 7 pair of contrasting characters. You can also write them as or 14 pure line. Right? Now what are those 7 characters? First is stem height, right? Second is flower color. Third is flower position. Fourth is pod shape. Fifth is pod color. Sixth is seed shape. And seventh is seed color, right? So these are the 7 pairs of contrasting characters which were chosen by Mendel in order to understand what is the pattern of inheritance of gene. And on the basis of these 7 characters, he basically performed 2 types of crosses by the name dihybrid and monohybrid. Now, on the basis of monohybrid cross, what results he proposed? Let's try to see. And before we do that, let's try to see first what is monohybrid cross. So, beta, when I talk about monohybrid cross, just a second, let's add a page. Okay. So, now, first, if I talk about monohybrid cross. Now see, mono means one, right? Cross means performing an experiment, performing an hybridization experiment, performing exchange of gamete. So basically here Mendel chose 2 parent. Parent has to be always pure line, one homozygous dominant and another homozygous recessive. This ma'am has already well explained in her class. So first and with the help of 7 characters which Mendel chose, he can obtain 7 different monohybrid cross, considering each character at each time. So when you start the cross, the first step is to select the parent. And now when you always select the parent, beta, then parent are always pure line. Right? So now if I choose my character, that is stem height, to perform monohybrid cross, now this stem height is controlled or it's governed by, suppose, T gene, right? So it is governed by which gene? It is governed by T gene. So what are going to be my parents, pure line parent? They are going to be homozygous dominant, right? And homozygous recessive. Suppose this is my tall parent and this is my dwarf parent. Now, first of all, these parents, they are going to perform reduction division. They are going to frame their own gametes. You know how to obtain gamete? The simple formula of obtaining gamete is 2 की power n. where N stands for degree of heterozygosity, right? So over here, when you see, you're only considering one gene and that gene is itself present in homozygous state. So therefore, over here, the value of N will become equal to 0 and anything raised to power 0 is actually equal to 1. So how many gametes will be obtained from both the parent? Only one type of gametes will be obtained. So this is going to produce a gamete with only one allele, that is dominant allele, and here a gamete is going to be obtained with the recessive allele. Now when these gametes are going to fertilize, a new offspring will arise which is referred to as first generation offspring, called as F1 generation offspring. So all F1 generation, their phenotype will be tall. Now, somehow Mendel got confused that why all offsprings are appearing tall, as you chose two parent where mother was dwarf and father was tall. So hasn't mother contributed any of her character? क्या सच में ऐसा है? अगर ऐसा है, then तो this is not a right pattern of inheritance. Why mother is not contributing? Why only father phenotype is accepted by the offsprings? So to conclude all these confusion, this fellow Mendel performed the next step of the experiment, beta, which is known as selfing. Selfing, right? So now what are we going to do in selfing? The F1 generation offsprings, they are going to be crossed with each other. Now again, first of all, you have to obtain the gamete. The simple formula of obtaining the gamete is 2 की power n. Now over here, if you check the parental nature, the gene nature, the gene is beta present in heterozygous state. Now if the gene is present in heterozygous state, then the value of n over here will become 1. 2 की power 1 means how many gametes will be obtained? Very good. We are going to obtain basically 2 gametes from each parent. And now you are going to draw the Punnett square to show the possible zygotic combination of these gametes. Right? So basically when he performed the Punnett square and when he saw, then his results were absolutely amazing, which proves the phenotypic ratio of F2 generation as the phenotypic ratio of F2 generation as 3:1, where beta 3 stands for tall and 1 stands for dwarf. You see over here, check in this Punnett square, you yourself will get the answer. These 3 offsprings whose genotype is either homozygous dominant or heterozygous dominant, then they are going to be tall. But this offspring who's having both the recessivity allele will be dwarf. So ratio is 3:1. In short, you can say if 100 offsprings are present, then out of which 75 will be tall and 25 will be dwarf. So now like this, it proves that none of the characters of the parent is lost. Even mother character of being dwarf is shown by some of the offsprings, not if in F1 generation, then of course in F2 generation, right? Clear to everyone? Okay, very good. So this is the phenotypic ratio. Now, suppose if you beta have to write the genotypic ratio for this, let's go back to the Punnett square and let's try to see. So how many are homozygous dominant? 1 homozygous dominant, 2 heterozygous dominant, and 1 homozygous recessive. So accordingly, the genotypic ratio will be 1:2:1. Mendel calculated this by simply performing test cross, right? So what are you going to write? By performing Yes, by performing test cross, we can calculate the genotypic ratio, which is 1:2:1, where 1 stands for homozygous dominant, 2 stands for heterozygous dominant, and again 1 stands for homozygous recessive. So this is going to be the answer, your phenotypic and genotypic ratio for monohybrid cross in their F2 generation. Now, with the help of this result, this fellow Mendel proposed two basic laws, out of which one law is purely and 100% accepted, but the second law, that is law of dominance, was not accepted, as there were so many exceptions to law of dominance. But this law of segregation, also known as beta law of purity of gamete, it is the 100% true law with no exceptions. And now what this law states, so basically beta, according to this law, if any gene has to be inherited from the parents to the offspring, then during the time of meiosis, during the time of segregation, both the alleles of the gene, they segregate, they separate out independently without influencing each other, and each gamete thus receives only one allele out of the two. For example, if T gene is present in heterozygous state, right? Now when segregation will occur, when meiosis will occur, so on reduction division, that is also called as meiosis, both the alleles will segregate out independently and each gamete is going to receive only one of the allele. So this is known as law of purity of gamete. Why purity? Purity stands for haploidy. That is, out of the two allele, every gamete is going to receive only one allele. Right? Clear? So now what are you going to write for law of segregation? Chalo, see everyone. So basically beta, according to this law, right, comma, according to this law, on segregation, on separation, or on reduction division, each allele of the gene segregates independently such that one allele out of the two out of the two is received by each gamete. Right? So this is 100% true law. However, the second law proposed on the basis of on the basis of monohybrid crosses, law of dominance, this was not a true law, which had so many exceptions, because of which later on this law got failed even. And now first of all, before we talk about the failure of law of dominance beta, you should know what is actually law of dominance. Correct? So according to this law, इसके हिसाब से, first of all, if a gene is beta present in heterozygous state, right? That is, if a gene is present in heterozygous state such that one will always be expressive, called as dominant, one will be always dominant, and another recessive to it. Right? Second point, according to law of dominance, beta, every gene is only represented by two allele, not more than two allele. Second point, a gene is only represented by two alleles. And third point he stated, according to law of dominance, every gene can only control one character. So every gene beta controls one character only. Okay? Now see over here, all the three statements proposed by Mendel in his law of dominance are wrong. Why? Because one gene, if it is present in heterozygous state, then maybe a dominant allele is not completely dominant over the recessive allele, and maybe both the alleles are equally expressing themselves and none is dominant or recessive with respect to each other, like you see in case of codominance and incomplete dominance, right? So incomplete dominance was then proved by Carl Correns while working on the flower color of Snapdragon, and according to that he proved first line of the law of dominance absolutely wrong. Then come to the second line. What does the second line says? It says that a gene is represented by two alleles. However, you know, it's not always true. A gene can be represented by even more than two alleles. This condition is known as multiple allelism. The best example to explain multiple allelism is your own blood group. Our blood group is represented by more than two alleles, which are two dominant allele, one recessive allele, IA, IB and small I. Right? Then third, he said that every gene is represented by, or every gene controls only one character at a time. This is also wrong. Why? Because you know the condition of polygyny and pleiotropy. Polygyny means, so what is the exception to this point of the law of dominance? Polygenic inheritance and pleiotropy. So everywhere there were lot many exceptions, and for so many reasons this law of dominance was then proved wrong and was discarded. Polygyny means that many beta gene can control one character, right? Whereas pleiotropy means that just reverse of polygyny, that one gene can control many character, right? The best example of polygenic inheritance is your own very human skin color, height, intelligence, etc. You must have seen that we have so many different shades of human skin color. Some are Negro, some are as white as Americans, and we Indians are neither too black nor too white. We have somewhat wheatish color. So all these different phenotype or different range of human skin color is actually a polygenic effect which is controlled by three pair of genes. And here number of dominant allele is responsible for producing the amount of melanin. So if you have more number of dominant allele, more melanin will be produced, and accordingly your skin color will become darker, right? And the example of pleiotropy are again so many. For example, there is one well-known autosomal recessive disorder by the name phenylketonuria. Now, basically, beta, this disease occurs when a specific gene present in the liver stops working. Now, when that gene stops working, as a result, a specific enzyme called phenylalanine hydroxylase production gets stopped. Now, since our liver cells or liver is not having the enzyme phenylalanine hydroxylase, which thus stops the conversion of phenylalanine into your tyrosine. Now, since there is no conversion of phenylalanine into tyrosine, so this phenylalanine starts converting into some other chemical like phenyl acetate, and thus it's this situation where conversion of phenylalanine into tyrosine stop causes mental retardation, causes your discoloration of urine, hair pigmentation loss, body hair loss, etc. And this is known as phenylketonuria. Right? Clear to everyone? So this is what Mendel's monohybrid cross is all about. Now, apart from performing monohybrid cross, in the same manner, beta, he also performed a dihybrid cross, where at a time he chose two characters for performing the cross, and then accordingly he obtained the results. Right? So next heading is going to be dihybrid cross. Right? Now dihybrid cross means a cross made by choosing two characters. Cross made by choosing two characters. Right? Chalo. So now let's see how these two characters, they are basically going to work. Now basically I'm going to portray one of the dihybrid cross over here, and then you yourself have to make out. So two characters which I am choosing is, for example, T, that controls the stem height, and R, which is responsible for controlling seed shape. Right? Now, so always whenever you begin the cross, always you have to first choose the parent, and your parent are always pure line. So over here, my pure line parent are going to be homozygous tall with round shaped seeds crossed with homozygous recessive for both the character, that is dwarf parent with wrinkled seed. Now, what is the first step? Obtaining the gametes. Now, over here, if you see, both the genes of my parent are present in homozygous state. So, the value of n in the formula will be 0. If the value of n is 0, then the number of gametes obtained will again be only 1. So, it is going to give 1 gamete. Same over here. Now, everything you just have to repeat same as that of monohybrid. Dihybrid is nothing. It is just the copy of monohybrid cross where you are considering one character more. Right? It's nothing. इसमें कुछ ज़्यादा नहीं है. इसमें आपको एक character से ज़्यादा, you have to choose one more character. You are basically, you are having over here two characters, and on the basis of that you are performing the cross exactly in the same manner as you were doing for monohybrid cross. Right? Clear? So now, simply after obtaining F1 generation, you are going to perform the selfing step, and on selfing, what are you going to obtain, beta? You're going to obtain phenotypic ratio as 9:3:3:1, where 9 are round and tall plants, 3 are round but dwarf plants, next 3 are wrinkled but tall plants, and one category is for dwarf as well as wrinkled seeds. Right? Clear? So this is going to be the phenotypic ratio, and the genotypic ratio you have to remember 1:2:12:24:12:1. So this whole is the genotypic ratio, beta, which also you have to remember. Now, on the basis of his monohybrid cross, he framed two laws. Now over here, on the basis of his dihybrid cross, Mendel framed one law which is known as law of independent assortment, right? So over here he basically proposed law of independent assortment. Now, what is this law all about? This is very important for you to know. It is just the combination of two laws of segregation. Now, here, what does this law of independent assortment means basically, beta? According to this law, whenever more than two genes are present on the same chromosome, right, then each gene on the same chromosome during the time of meiosis, they segregate first from each other independently, without influencing, without blending, and then followed by their respective alleles. It's very simple to say. Suppose this is my chromosome, right? On this chromosome, I have two genes, right? Now, this is the situation which I have drawn in simple manner. Now, if my both the genes, now meiosis will happen. Now, due to meiosis, both the genes will segregate. So, according to Mendel, whenever both the genes they segregate, then they segregate independently without influencing each other, like this. And then their alleles also segregate without influencing each other. So this segment is okay. See students, this segment is true. This is law of segregation. But this segment is wrong because there are possibilities of linkage. Right? So what is linkage basically? Linkage is when two genes located on the same chromosome, less distantly or close to each other, they tend to get they tend to get along with each other, and because of which their separation is not 100% according to Mendel's law of independent assortment. So linkage basically becomes the exception, right? And this linkage term was coined by Morgan. Correct, everyone? This term was coined by Morgan while working on fruit fly, which is scientifically known as Drosophila melanogaster. This organism is having very good features. This Drosophila, which is commonly called as fruit fly. It has so many good properties because of which it is considered as one of the best organism to study. Right? Clear to everyone? Now, like for example, it's weak. Its lifespan is just limited to 2 weeks. There's a very good sex determination simply by looking at there's a clear sexual dimorphism exhibited by Drosophila. You can identify male and female simply by looking at them. How? By simply males are smaller in size as compared to female. Then they are very easy to grow. They can grow on any simple synthetic medium. Per mating, they produce large number of offsprings, and their pattern of sex determination is also same as that of humans, which is XXXY type, right? So because of all these good properties, beta, Drosophila is considered as the best organism to study gene related researches, and that is why Morgan chose this and coined the term linkage, right? Now what basically linkage is, now that I am going to tell you. So let's see what actually is linkage. Linkage is beta whenever two genes are closely present on the same chromosome, right? Whenever two genes are present closely on the same chromosome, they do not, Mendel said कि they will, वो करेंगे segregate, but the truth is they will not segregate independently in that case. So they, two genes are closely from the same chromosome, they do not tend to separate independently, beta, rather they get linked. Now, as a result, now because of which what will happen? Yes, very good. As a result, chances of crossing over will decrease, because of which percentage of formation of recombinant gamete as to that of parental gamete will also fall, right? So, as a result, percentage of crossing over decreases. Hence, percentage of beta parental recombinants increases, right? And percentage of recombinant decreases. Correct? Yes or no? And then there was a student of of this fellow Morgan by the name Alfred Stewart Went, who by using this concept of that percentage of crossing over is directly proportional to the distance between the two genes. More the distance, more is the crossing over. More less is the distance, less is the crossing over. So according by using this law, one of the student of Morgan by the name Alfred Stewart Went tried to locate the genes on the chromosome, stating that percentage of recombination will be equal to the distance between the two genes. Yes, have you all understood till here? Okay, well done then. So now this point also let's write on the screen that Alfred Stewart Went located genes on chromosomes by stating that percentage of crossing over is equal to the distance between two genes, and unit will be cm. Right? Correct, everyone? Now, moving towards the next important topic of the chapter, that is mutation. So, how will you define beta mutation? Mutation is basically sudden, maybe inheritable, maybe not inheritable, change in the genotype of an organism, because of which the phenotype of an organism also changes. The term mutation was coined by Hugo de Vries while working on Oinothera lamarckiana. Basically, beta, mutations, they can be of three types: gene mutation, genomatic mutation, and chromosomal mutation. Correct? So, how will you define mutation? Mutation is nothing but simply sudden change in the genotype of an organism because of which the phenotype also changes. Right? And this can be brought by three well-known reasons. The three well-known reasons are, gene mutation, genomatic mutation, and chromosomal mutation. So how many types of mutations are possible? Yes. So there are three possibilities: gene, genomatic, and chromosomal. Right? Gene mutation means when a particular gene and their associated nucleotide shows some change, like for example in sickle cell anemia, so that is gene mutation. Genomatic means then chromosomal means that on the same chromosome a number of genes get altered but without changing the total set. In genomatic, either whole chromosome get deleted or added up, or whole sets get deleted or added up. So accordingly, these are the three types of mutation. I hope you all are well aware of this. Ma'am has already taught well in her classes, right? Now, the term mutation was given by Hugo de Vries, right? Now, moving towards the next very important topic, that is sex determination, which was proposed by Henking. So what Henking has to say? So according to Henking beta, he basically studied spermatogenesis in insects. Spermatogenesis in some insects, and he observed that only 50% of the gametes were receiving the X chromosome and 50% of the gametes were not receiving X chromosome. So not later on, this another chromosome was referred to as Y. Right? Now, the basic method of determining sex is genetical, which can be divided into two categories as male heterogamety and female heterogamety. Male heterogamety means males are responsible for producing two types of gametes, like you see XXXO type and XXXY type, whereas female heterogamety means where females are responsible for producing two types of gamete, like you see in case ZZZW and ZZZO type, right? Very important, students, you all have to remember this thing. So there are two ways to determine sex. One is referred to as male heterogamety and second is referred to as female heterogamety. Male heterogamety are of two types, XXXO type, XXXY. We fall in this category along with Drosophila. Female heterogamety is of two type beta, that is ZZZW type, ZZZO type. Yes, right? So this type of sex determination is known as chromosomal or genetical sex determination. Correct? Now, moving towards the next topic of the chapter, which is pedigree analysis. Basically, beta, when it comes to pedigree analysis, this is generally helpful in those or to those people who are expecting their child in order to detect that whether their offspring is going to suffer from a specific disorder or not. So basically, pedigree analysis helps to detect the possibility of any disorder passing passing from generation to generation. Passing from generation to generation in a family. Then there's whole pedigree analysis. There are whole pedigree charts. Accordingly, you solve it. There are two types of disorders basically. Disorders can be divided into two categories as Mendelian, which can be traced in pedigree or which you can study, and second are called as chromosomal, that mainly arises due to mutation, right? Mendelian are again of two types, autosomal and sex linked. Autosomal basically beta means when the disorder is present on autosomal gene, right? Autosomal gene disorder. And sex linked means when the disorder is present on sex chromosome, like hemophilia and color blindness are sex linked recessive disorder, whereas your phenylketonuria, sickle cell anemia, thalassemia, cystic fibrosis, all these are myotonic dystrophy. All these are basically beta autosomal disorders, may be dominant, may be recessive, right? Clear? In chromosomal disorder, you have like Turner syndrome, Klinefelter syndrome, and Down syndrome. Down syndrome occurs due to the trisomy of 21st chromosome. Klinefelter syndrome occurs due to the trisomy of sex chromosome in males, and and this Turner syndrome occurs due to the monosomy of sex chromosome again in female, right? So this is all about the recap of this simple chapter, that is genetics. This whole was a very simple conclusion of the whole chapter. This recap was really a quick recap. I hope you all have just got the gist of your NCERT textbook. For more details, go through each and every lecture of the class, which is really going to be very, very helpful for you. If you have already learned all the chapters, all the lectures of this chapter, this recap is again going to be fruitful for you. Now, my only homework for you all will be to revise the chapter from your main notes that is given to you in your classes. Plus, you also have to go through all the DPPs, you solve them, plus all the test series, always be present in every test series. And of course, studies has to be, studies are very, very important beta, not only classes. If you're regular in class, it's very good. But suppose if you're not studying at home, if you're not giving time by your own, then again all these things will be of no use. Till the time you don't have a specific timetable, till the time you're not devoting your time to your self studies, till the time your doubts are not getting clarified, till the time you're not active in your test sessions, it's not going to help you. So make sure that you have a proper timetable, you are following it. Then you should also give more time to your self study, then appear for the test, solve the DPPs given to you on daily basis, right? Chalo. So with this, I would like to end my today's ultimate recap session. I hope it was enough and fruitful for you all. Thank you for joining me. See you in the classes. Till then, bye bye everyone. Take care.